Before establishment the primary culture, we put the necessary reagents into the incubator to warm them up. Then the reagents heated in this way are transferred to the laminar chamber. According to the procedure, before starting work we disinfect the gloves and the working surface of the laminar chamber worktop with ethyl alcohol in order to minimize the risk of contamination. Prepare a petri dish and add a set of enzymes on it consisting of collagenase, pronase and trypsin. The falcon tube contains a fragment of the cancerous tissue of the patient's lung immersed in a solution of saline and antibiotics. We transfer a piece of tissue to a petri dish so that the enzymes can start working. Then we fragment the tissue into smaller elements, about 1 to 2 millimeters, in order to increase the surface area of enzyme activity on the tissue. Lung neoplastic tissue is often quite fibrous, so choosing a scalpel for fragmentation is not the best option. Small scissors work best. The added enzyme mixture in this case is to break down the neoplastic tissue and detach individual cells from the compact mass of the tumor, from which the primary culture will later be established. Collagenase is designed to digest collagen, while trypsin digests glycocalyx and extracellular matrix proteins. Pronase is here an enzyme supporting the activity of the two previously mentioned enzymes. As we can see, fragmentation is quite a tedious and time-consuming process. However, its correct and accurate performance guarantees a high recovery of cells, which will later be used to establish a culture. The more finely fragmented the tissue, the greater the cellular recovery will be due to the increased surface area of enzyme action on the tissue. Here we see the fragmentation process in close-up, and we can of course see the approximate size of the fragments into which such tissue should be fragmented. After tissue fragmentation, the Petri dish should be transferred to an incubator because at 37 Celsius the enzymes work more efficiently. Before moving the dish, it should be closed and secured against contamination. For this, we will use parafilm, which is a semi-permeable membrane. Put the parafilm around the Petri dish so that it fits tightly to its edge. Then the prepared and secured plate is transferred to the incubator for about 15 minutes. The time of enzymatic digestion should not be too long to avoid destruction of the cell membranes by the action of enzymes. Taking advantage of the moment of free time, we can prepare the cell culture medium necessary for our established culture of cells. We will prepare the culture medium in a 50 ml Falcon tube. And the first component of the culture medium will be DMEM, which is basic component. DMEM contains all the essential amino acids, vitamins and minerals. Its red color is due to the presence of the pH indicator phenol red. This indicator allows to determine the sweetability of a medium. A color change from red to yellow suggests that the medium should be replaced. The differences in the colors of the media can be observed in the present photo. The color change associated with the change in pH is caused by the secretion of acid metabolites by the cells during their life. Another very important component of the medium is FBS, fetal bovine serum. It provides cells with hormones that stimulate cells' growth and adhesive factors. In order to reduce the risk of possible contamination, mixtures of ATB antibiotics in the amphotericin, streptomycin, and penicillin set are added. The last components of the medium is a mixture of insulin, transferrin, and sodium selenate. Insulin is necessary for cells to nourish. Transferrin is a carrier of iron needed for cellular respiration, while sodium selenate is a free radical remover and prevents against damage to genetic material. We close the Falcon tube containing a ready medium and we will use it later in our further work. After about 15 minutes, in which we have prepared a culture medium, transfer the petri dish with the fragmented tissue left in incubator back to the laminar chamber. Remove the parafilm, which, as you can see, is firmly attached to the edge of the petri dish. We will collect the cell suspension, from which we will cultivate into a 15 ml capacity Falcon tube. To make it easier to handle only the liquid with suspended cells, some tissue fragments are removed with a scalpel on the dry surface that was created after tilting the plate. We will trying to collect only the cell suspension. Of course, it will probably happen that an unwanted fragment will be transferred into the pipette pit, but we will cover that in a moment. After transferring the liquid to the falcon, wait a moment until the randomly taken fragments fall to the very bottom. Then we transfer the cell suspension from above the fragments that have fallen in the sedimentation process to the new falcon. Then the falcon with the cell suspension is taken to centrifugation in order to recover cells from the suspension. The observed photo shows that the enzyme mixture changed its color from transparent to cloudy brown, which indicates the presence of a large number of cells in the suspension, which, due to the enzymes, were separated from the compact mass of tumor tissue.